So welcome to our seminar for this week. This is Christine O'Connell, who just nicely introduced herself to everybody in the room. If you're not in the room, you missed out on a handshake and a hello. Christine has some history here at U of M with her PhD, and then was at Berkeley for a postdoc, and now is back in the cities at Macalester. So I'm sure she'll give us a lot more details about that trajectory, and she's going to talk to us about soils in Puerto Rico and some big disturbance events. Thanks.
Awesome. Yeah, thank you for that introduction. Thanks for having me, everyone. So, Rachel, if there's any comments that are like, this is too quiet, just give me a flag. This is pretty small. Oh, yeah. Oh, awesome. Cool. Yeah. So, but if you see any comments about technical difficulties, like, don't be afraid to stop me. Oh, thanks. Let's see. Okay, I'm about to risk it all by unplugging and plugging back in because I think I need to not mirror screen. Let's see. Extend desktop. Let's try that. Okay. Hopefully that will work. Well, oh shoot.
You could probably like unshare the screen and then reshare it and then just click the like different monitors, if that makes sense.
Ooh, that's a good idea. Ooh, did that work? Thank you. Brilliant. Although wait, you guys are still seeing the wrong screen. Uh-oh. Yeah, let's see. Now I think if I mirror display. No, this is worse. I'm gonna give up after this, but we're gonna try one more thing. Let's see. All right, any thoughts? You guys want me to just stick with this? Should we sync a bunch more time? Okay, online people can see, you guys can look over here. I appreciate your patience. Sorry about that. Okay, well, anyway, thank you for the intro. My name is Christine Sierra O'Connell. My pronouns are she, her, and Aya. I'm a faculty member at Macalester College. And today I'll be talking quite a bit about a bunch of the work that my lab group is doing, looking at disturbances and how they're altering tropical biogeochemistry. But usually when I give talks outside of small liberal arts college contexts, I also like to super clarify the context of my job because I find often students and postdocs are really interested in what a career at a primarily undergraduate institution looks like. So yeah, a big part of my job is doing really cool research science. We do quite a bit of science and research at Macalester, but with an eye for providing growth opportunities to early career scientists and undergrads. So I have a photo here of my amazing lab group from this last summer. They're going to be featured quite a bit in this seminar. So I'll just point them out now. This is Rachel Cruz Perez. She's a PhD student at Penn State who was working with our group. She's really interested in kind of mentoring undergrads, which is how she got involved. And these are three Macalester undergrads, Trung Dam, Nivia Becerra-Santillan, and Miriam Pleat, all three of whom are kind of like really early career scientists. And we often take on scientists like this, not only to send them on to do master's and PhD degrees, but also to kind of like gain technical skills before working for government or environmental consulting. We have those sorts of pathways really commonly throughout our undergraduate population too. So I'm very open to answering questions from any of you or your students or mentees about kind of like what this career shift looks like. It's a very different way of being a kind of working scientist and educator, but it's really fun and we actually do a lot of really cool stuff too. So yeah, that's kind of the context. All that being said, let's talk science. Okay, so my lab group works on the feedbacks between climate change and terrestrial ecosystem processes.
I'm so sorry.
No.
The slides are not moving on.
Okay, great. This is actually great because this is bothering me too. Okay, should we just like,
Let's go ahead and
let's see.
do it how we had it before, where they would see the.
Okay, so we're back to extending desktop.
Mm-hmm.
I'm so glad you stopped me. Oh wait, swap displays.
Your screen.
Okay, so now we're seeing the right thing.
We're seeing the right thing, but they're not seeing anything.
Oh, my screen's not shared at all.
Yeah.
Gotcha.
So desktop 2.
Okay. Thank you for your patience, everyone. You would think we hadn't all taught online on Zoom for a year and a half, and yet these skills are like gone forever. Okay, is that okay? Let me like click one back. Did that move? Okay, awesome. Okay, so what does my lab group work on? Geez Louise. Oh, crazy. All right, well, sorry about this too. Okay, so what does my lab group work on? My lab group works on the interface of terrestrial ecosystems and climate change and asks how global change and global change disturbances are shifting these dynamics between terrestrial biogeochemistry and atmospheric dynamics and climate change. So broadly, we look at this. Climate change is exerting influence down, metaphorically speaking, onto a terrestrial ecosystem. That terrestrial ecosystem is experiencing subsequent changes to the carbon, nitrogen, and phosphorus cycles. And then those changes are feeding back to the atmosphere. All of that is happening while global changes are also shifting greenhouse gas emissions directly and indirect shifts to species composition, either between land uses, across systems, or because of these disturbances. So there's sort of like many different indirect feedbacks that might shift biogeochemistry, and also many different pathways through which the carbon, nitrogen, phosphorus cycle can shift either water quality or atmospheric climatic dynamics. And as I mentioned before, we're doing this with a whole host of undergrads who are being introduced to science. And not only are we trying to provide them with growth opportunities, we're also trying to model what it looks like to do ethical science in kind of a diverse, modern, DEIJ-informed context. And the reason that that's so front-loaded in my group and at Macalester in general is because we do a lot of work internationally. So this is a photo from the annual meeting of a project I work with that we'll talk about later in the seminar called TRACE. TRACE is based at the International Institute of Tropical Forestry in Puerto Rico. So we do a lot of interfacing with Puerto Rican scientists outside of our immediate local context. And we do a lot of thinking about how to do that work well without being helicopter scientists, without kind of extracting intellectual capital from the environments that we're doing the science in, and how we're also having like really strong back and forth with these collaborators and making sure that we're giving as much as we get from these relationships. Okay, so back to what we work on, this interplay between disturbance, terrestrial biogeochemistry, and climate dynamics. So in this talk, I'll be talking about a few different types of disturbance. So I'll just center that for us really quickly. The first is that we're going to be thinking about precip. We know that precipitation changes to timing and amount are already ongoing and likely to become more expansive and more severe over time, depending on where you are on Earth. So we're going to be kind of having like water cycle precipitation in mind. Where I work in Puerto Rico and in the neotropics is a very heavily disturbance and hurricane tropical cyclone impacted area. So what we're looking at here is a map of the historic cyclone tracks from NOAA. Puerto Rico's right over here. And you can see this is a system that is very impacted by hurricane events. So the primary dynamic that's being shifted because of climate change isn't that hurricanes ever come to the system. It's that the more extreme hurricanes are coming at a faster turnaround rate. So we'll get to this a little bit later in the talk, but very broadly, our rule of thumb right now is that in the Caribbean and much of the neotropics, severe hurricanes, so category 4 or 5 hurricanes, depending on the model you're using, are probably returning about 2 times as quickly as they were previously. So for instance, in this system in the 60s and 70s, large hurricanes probably had a return rate of about every 60 years. Now really large hurricanes are probably returning every 20 to 30 years. And you can imagine that that trend might continue to accelerate over time. Okay, finally, we're not going to focus on that much today in this talk, but my group also does quite a bit looking at land use change. My PhD at the U was exclusively focused on land use change. And even though I've gotten away from that work really broadly, I've started to use that a lot in the context of being at a primarily undergraduate institution, because a lot of our students want to do super applied work that's super local. So I've ended up shifting a big part of my research program to extremely small scale, hyper local, hyper applied, like some people would almost call them activist science projects. And they're exclusively student led, and they're really, really fun. And it's kind of like a very different way to think about using your scientific toolkit. So I'll touch on one of those projects at the end to just kind of like show what it might look like to do this kind of work at a school like Macalester. Okay, so what are we talking about today? We're going to go through 3 core questions, sort of 2 big case studies, and then a little dabble at the end. The first question is going to be asking about how drought is feeding back to the climate system in this forest in Puerto Rico that I mentioned before. The second is how chronic warming in this forest is going to interact with acute disturbance events, and kind of how we're thinking about measuring and modeling those interactions. And the last is just thinking about how to use these sorts of toolkits to advance land use sustainability, especially with kind of like really applied science outcomes in mind. Okay, so first big question: how does severe drought impact below ground nutrient cycling and greenhouse gas emissions? So what you're looking at here is a photo from El Yunque National Forest in Puerto Rico. I'll show you on a map in a second, but this is an experimental forest. So we have many decades of data and a lot of information about the system. So it's a really great case study location to ask questions about how kind of like shifts in global change drivers over time might be shifting the system, because we actually know a lot about how the system used to work in comparison to some of our kind of like peer forest sites. Okay, so the first thing I want to note is that this question is kind of influenced by the fact that this is not a very droughty system in general. However, in 2015, there was a lengthy and severe historic drought. So this is data from that drought. What you're looking at here is on the x-axis, January to January, so generic date. And on the y-axis is the daily precipitation anomaly for 2015 versus the preceding 10 years. So if a given bar is red and below the 0 line, that's like saying that on this date, February 12th or whatever, February 12th in 2015 received this many millimeters less of rain than the average February 12th in the 10 years prior to the 2015 February 12th. So the first thing I want you to note is that this drought was extremely persistent. It basically lasted the entire year. It was a really deep drought, it was really long, and it did not let up. Broadly, this system received about half of the amount of rainfall that it did in a non-2015 drought year. And it's a little bit of an interesting case because in comparison to those of us who also work in the upper Midwest, this was still a really big rainfall year. Like this forest received almost 2 meters of rain this year, but this is a forest that usually receives 4 meters of rain. So 2 meters of rain sounds like a ton, but if you're cutting your usual rainfall in half, that obviously has really big knock-on effects. Okay, so while that drought was occurring, my postdoc lab and I had an ongoing experiment that kind of allowed us to opportunistically track a bunch of what was happening before, during, and after the drought. So the experiment that we had set up was initially asking some more basic biogeochemical questions about topography. So the way it was set up is we had a ridge, slope, and valley that functionally acted like a sensor array. So along that ridge, slope to valley, which is a very common topographic gradient in this system, what we had was a series of transects with soil sensors looking at soil moisture, soil oxygen, and soil temperature on the hour. So 1, 2, 3, 4, 5, 6, 7, sort of 7 topographic zones, and then 5 repeats across space. So we basically ended up with this topographic gradient of soil moisture, temp, oxygen from a ridge on a slope and down into a valley. Then overlaid over that, we had a series of automated greenhouse gas sensors that were running to a Picaro that allowed us to measure soil greenhouse gas fluxes of CO2, CH4, and N2O. So that's carbon dioxide, methane, and nitrous oxide. Now, this let us spit out an insanely dense amount of data, which ended up being extremely powerful to track what the consequences of this drought were over time. That's going to be a bunch of the results I'm about to show you. And then over top of that, these boxes are quadrats we use to collect repeated soil cores. So we also had a bunch of kind of direct soil measurements of phosphorus cycling, nitrogen cycling, et cetera. Okay, here's a photo of the forest just to kind of center you into this kind of landscape. So things to know about this forest. It's dominantly aseasonal. So there is a bit of a dry season, but kind of on the global biome scale, this is a wet tropical forest that does not functionally have much of a dry season. Like I said, it's pretty topographically heterogeneous. We have ridge, slope, and valley. It's super biodiverse, relatively carbon rich, pretty tall statured, lots of palms, as you might be able to see. This is a pre-hurricane photo. Hurricane Maria that we're going to talk about later in the talk massively shifted this forest. But this is kind of like the baseline. And you can kind of see this is the experimental setup, tons of greenhouse gas measurements. We have lots of sensors here. And like I said, we were taking a bunch of other samples too. Okay, so these are some of our results from the 2015 drought. What you're looking at here is instead of a generic January to January x-axis, this is November 2014 into early 2016. So this is our pre-drought period, our post-drought period, and then this was kind of like what we were observing in the middle. In this particular graph, on the y-axis is soil moisture, and the colors are where you were on that ridge to valley gradient. So the cooler colors were up on the ridges, kind of that middle ground, oranges were on the slope, and this bright red was in the valley. So I want to point out a few things first. So these dashed lines were not a priori decided by us. We used a modeling technique called structural change modeling to basically ask, how does this data change in relationship to itself over time? And it's a really common technique used with time series data to basically try to break up time series into like functionally different periods. So our structural change modeling found that there wasn't just kind of a pre-drought and a post-drought period. There was a statistically distinct drought recovery period that functioned statistically differently than the pre-drought and the post-drought. So one of our first big takeaways was noting that when there was drought onset, there was an extreme threshold effect in which soil moisture plummeted basically everywhere, even in valleys. So these valleys where there's usually quite a bit of standing water, those got drier too. So big threshold effect with the onset of drought. This dashed line roughly correlates to when precipitation began again at roughly around the end of 2015. But you can see there wasn't that threshold recovery of soil moisture. So one of our other big findings was that there's a really long recovery and lengthy persistence of the below ground consequences of a big drought like this. And in fact, when you take these modeling results, if you only measured the end of drought by around when precip came back, versus when soil thinks that drought is over statistically, the drought was actually about 63% longer than you would imagine just from the rainfall record alone. So kind of one of our big results here was that from an ecosystem perspective, when a drought ends, might be very different for below ground functioning than from what we might think walking around with just like our low-hanging fruit data. Okay, another thing we were really interested in was oxygenation and how that might shift kind of microbial communities and greenhouse gas trace fluxes, or sorry, trace greenhouse gas fluxes. So what you're looking at here, same exact time series, these dotted lines are the same, pre-drought, drought, drought recovery, post-drought, colors are the same, ridges up top, valleys in red, but instead of looking at soil moisture, we're looking at soil oxygen. So this shouldn't be very surprising based on our moisture results. So you see a big threshold effect in which soil moisture goes down at the beginning of drought, gets drier, oxygen levels increase quite substantially, especially in valleys. And then you can also see that these oxygen levels take a while to recover, which sort of tracks with that moisture. But I think one other thing that we wanted to note is that even in this period where, according to soil moisture, drought has ended, you can see there's really big distinctions between soil oxygen values post-drought as there were pre-drought. So pre-drought soil oxygen values were way flashier, especially in slopes. There were big hot spots and hot moments of low oxygenation levels, and that kind of didn't really exist other than in the valleys in this post-drought period. So that's something we've gotten really interested in and we're doing some follow-up work on actually. Okay, so that's kind of the abiotic conditions. What does that mean for carbon, nitrogen, phosphorus? So what you're looking at here is a graph of pre and post-drought soil samples in which we measured inorganic phosphorus. So this is phosphate, that's the good stuff. Microbes, roots can take it up very readily. And what you can see is that no matter what topographic position you were in, after the onset of drought, inorganic phosphorus availability statistically decreased by a substantial amount. So very significant drop in the availability of this labile, super uptakeable phosphorus. At the same time, organic phosphorus, which has yet to be broken down by microbes and isn't readily available to plants, went up. Okay, great. So part of our story here is that probably because of changes to decomposition rate, maybe because of some changes to iron cycling, inorganic phosphorus suddenly became relatively unavailable, and organic phosphorus started to kind of like build up over time and not get processed as readily. Okay, so that's a pretty interesting geochemistry result. But if you kind of like take a step back as an ecologist and think about plant communities, there are actually probably a bunch of really important knock-on indirect effects of this. So for instance, in the immediate wake of a major stress like a drought, many tree species and individuals drop a bunch of their leaves. They change how they're allocating their carbon resources. They shift kind of the level of dormancy. They're more susceptible to disease or kind of like microbial attack, et cetera. So usually in the wake of drought or any other similar chronic disturbance, those trees are kind of like greening out, leafing out, trying to do a bunch of photosynthesis, get a bunch of new carbon, and either grow back tissues that they dumped when they were trying to survive at the beginning of drought, or they're growing more roots to scavenge for more things, they're repairing damage, they might regrow other tissues, et cetera. So that's suddenly going to be a lot more difficult if you don't have one of the core limiting nutrients available to you as much as you did before drought. So one of the projects that we're working on with some collaborators now is trying to have a better handle on whether or not this shift in phosphorus availability has a long-term potential impact on carbon storage in the system. So like one question you might imagine asking is, if these droughts become more frequent and more severe, and it's slower, the plant community is slower to recover because of a loss of phosphorus, does that mean that the overall above ground carbon storage in plant tissues is going to go down over time? And I think that's a very open question that, like I said, we're trying to track down. Also, kind of a lot of remote sensing scientists are working on, and other places that have experienced tropical drought in the last 15 years. And I think this kind of falls into the bucket of like, this probably isn't great, but we don't totally know how bad it is. Okay, so that's kind of like big finding number one, these indirect effects from phosphorus cycle changes. We also directly measured what was happening to the carbon cycle via shifts in soil greenhouse gas emissions. So here you're looking at a slightly different type of bar graph. Instead of topography on the x-axis, we're back in that time series, pre-drought, drought, drought recovery, post-drought, and color is whether or not these measurements were from a ridge, a slope is purple, a valley is red. This graph is CO2 flux rate from soils on the y-axis. So these are coming out of these automated soil chambers. And I guess one piece of context here is that because we had these automated soil chambers, this technology is relatively new, right? This is all spun up in the last 15 years, and it's allowed us to have an amount of data that makes us pretty confident in these results. And it's actually like been kind of amazing to be able to mine these data. Like in my PhD, I measured a lot of greenhouse gas fluxes from static chambers. I feel like I worked really hard, and I had like 500 measurements. And I was like, wow, I did such a good job. In comparison, we had 10,000 measurements across this time series per gas. So we actually had 30,000 measurements, which like makes you feel like your PhD is like so dwarfed immediately. But it's like such an amazing tool to have access to these new instrumentation methods. Okay, so some things to point out here. Pre-drought, we had a CO2 soil flux pattern that we were pretty much expecting. Valleys are wet, they're relatively low in available oxygen, so they don't emit a lot of CO2. Cool, that story definitely tracks. Ridges and slopes are dumping kind of a lot of CO2 into the atmosphere. They're doing respiration at a pretty high rate, totally expected. Okay, with drought, one of our big findings is that valley CO2 emissions went up. That makes sense. Moisture went down, oxygen went up, CO2 emissions also go up. We also saw a big increase in CO2 coming out of these slopes. Okay, so drought recovery and post-drought is where we found a bunch of things that we were not expecting to. So the first thing that we were expecting is we thought that post-drought, when soil moisture and soil oxygen stabilized, that our soil CO2 and CH4 stories would stabilize too. But instead, we found that valleys continued to dump a ton of CO2 into the atmosphere. This was pretty unexpected. We also found that slopes recovered to their pre-drought emissions rate, but ridges continued to emit kind of a lot of CO2 and in fact, a statistically higher amount. So basically one of our big stories is that post-drought, even though we had this kind of below ground recovery, probably because of that like lack of jumpiness in O2 availability, we still saw a lot more CO2 coming out of the system post-drought than pre-drought. Okay, when it comes to methane, we also kind of saw an expected story at the beginning and an unexpected story at the end. So pre-drought slopes and ridges net take up a little bit of methane. Sometimes during some parts of the year, they're roughly neutral, but broadly in these sorts of tropical systems, upland soils are often a net sink for methane. So that's what we saw in the upland soils. And then we saw valleys dumping tons of methane out. Not surprising, these are wet, they're low in oxygen. We know that in general, these tropical wet, warm valleys are methane hotspots. Great. So after drought onset, when valleys get a lot drier, they suddenly see a huge savings in CH4 that's emitted out. And then our sinks of methane became stronger sinks. So we had a bunch of extra methane getting taken up in ridges and slopes and much less methane getting emitted by valleys. So far, if you stopped measuring here, you might be thinking, hey, maybe this is one of these rare negative climate feedbacks where climate is shifting, it causes global change, it then feeds back to the system to lower greenhouse gas forcing, which might make the next drought a little bit less likely or happen a little bit more in the future. Possibly a slightly positive ecosystem buffering climate story. But instead, we kept measuring. Drought recovery looked okay, and then post-drought, methane was just getting dumped out of the system. We were very surprised by these results. We were not surprised by the valley results. Valleys went back to being big methane hotspots, totally expected, totally normal. We were not expecting ridges and slopes to suddenly start dumping a ton of methane into the atmosphere. So this is an area that we're actually also doing a bunch of follow-up work on. Our working hypothesis in the paper was basically that decomposition perhaps had gone down so much during the drought that there was just an abundance of substrate for any microbes to work with. So that kind of tracks with the fact that we also saw a lot more CO2 coming out of the system, that maybe there's just like tons of carbon. Microbes were suddenly happy and they're just like metabolizing like crazy. And that the high rates of CH4 emission might be due to just like micro deoxygenated soil pores, maybe. But we also have a couple of hypotheses about how soil structure might have changed to maybe shift kind of the range of soil pore space and what the hydrology of the system might look like to make methane hotspots more likely in these upland sites. So I'll talk about that in a second when I kind of like trace down what we're doing next. But yeah, so this paper has led us to having a bunch of new hypotheses, and alongside some modelers, some climate modelers that we work with and that broadly work in this part of Puerto Rico, the entire LTER system in this forest has gotten really interested in drought. Because we know that the system is probably really affected by drought. It's not very evolutionarily set up for drought. There are clearly a lot of effects. There are a bunch of papers that came out of the 2015 drought, also looking at changes to the plant community and kind of like plant physiology in the system. So one thing that the LTR is doing is starting this year, the LTR, the Long-Term Ecological Research Station that is in El Yunque National Forest and that this research was based at, is running a new drought manipulation. So this is a photo not from the new manipulation, but from kind of a peer experiment in Panama at BCI. So basically our experimental manipulation is going to be an under the canopy rainfall exclusion experiment. We're currently in the 1 year of pre-treatment data collection. And yeah, I mean, basically we all have a million hypotheses of why what happened in the 2015 drought happened. And this is our kind of chance to manipulate a drought so that we can proactively measure things like soil structure before and after, kind of like run down some of these questions about, decomposition rates, et cetera. And there is a lot of interest in kind of like plant physiology as well. So where this project is going next is right now we're taking a bunch of greenhouse gas and soil structure and phosphorus and nitrogen measurements during our pre-drought year. And then we're going to kind of begin this drought manipulation experiment. And our main questions are mostly going to be, hey, how come a bunch of extra methane came out when we weren't expecting it to? And B, kind of what are the consequences for plants of these shifts in phosphorus? So yeah, some of this work, I want to give a shout out to Rebecca Montgomery. She's one of the main collaborators on this project on the plant physiology and community ecology side. Sampling began last fall. We have lots of data to collect. We're very open to collaborators, like come chat with me if you have any interest. And yeah, I'm always open to collaborating with new people. Okay, so the first big takeaway from case study number 1, drought, what does it do, is that for big disturbances like this in this system and probably many other systems, it's going to be really important to be thinking about both the direct effects and the indirect kind of like knock-on domino effects of a global change experience like this drought. So in this particular system, we saw this play out as direct effects of the carbon cycle through CH4 and CO2 emissions. But we also saw this play out as big shifts in iron and phosphorus cycling, which are probably going to have knock-on effects to carbon cycling as mitigated by plant community changes. Yeah, so to me, one of the big takeaways I've taken away from this project is that, like, my role as a field biogeochemist and ecosystem ecologist is probably to also have really strong connections to collaborators in modeling, both kind of on the microbial side and on the climatic side, because it's probably going to be really hard to get at some of these relationships if you're not able to use a bunch of different tools that can span across times. Okay, my second big case study before I talk about just sort of like dabbling in some applied science involves a bunch of undergrads. So this is kind of a great way for me to talk about all the ways we can work with undergrads really aggressively at a school like Macalester. So you'll be seeing like a bunch of undergrad faces and a bunch of undergrad results. I'm so proud of them. Okay, so our big question for case study 2 is how will chronic atmospheric warming, so a chronic disturbance like ratcheting up air temperature over time, how will that interact with acute disturbances? And we're specifically going to talk about the acute disturbance of a hurricane or the acute disturbance of a changing precipitation regime, and how will that interaction between these chronic and acute disturbances influence ecological patterns? Okay, so to study this question, we're going to, instead of looking at kind of like an observed 2015 drought, we're going to dive into an ongoing, proactive, large ecosystem manipulation experiment that I've been lucky enough to collaborate with. So this is a photo from the experiment I mentioned before called TRACE. TRACE stands for Tropical Responses to Altered Climate Experiment. It's a manipulation warming experiment. I'll show you a few more photos in a second. And just to orient us, TRACE exists here on the eastern side of Puerto Rico in this protected area. Okay, so this is a photo from TRACE. These are the same undergrads, Chunda, Miriam Fleet, maybe Becerra Santillan, and myself, who spent last summer down at TRACE collecting a bunch of data. So I like using this photo because my undergrads look so happy, but also this is one of the TRACE plots. So the way the TRACE plots work, I'm going to toggle back and forth between 2 slides for a second. This is an overhead image. There are 6 replicate plots, 3 that are control plots and 3 that are experimentally warmed. And the experimentally warmed plots are warmed using infrared heaters that are below the canopy level but above the understory, and that are warming the warm plots 4 degrees Celsius above the dynamic ambient plots. So basically, there are temperature sensors in the ambient plots getting routed into a dynamic system that says, okay, the ambient plots are currently whatever, 23 degrees C. So the warm plots need to be given X amount of electricity to get to 27 degrees C. And this system is pretty robust. So there's a pretty consistent +4 degrees treatment over time. That said, the treatment did fall out for a couple of months in the immediate wake of Hurricane Maria, but I'm not going to show you any results from that kind of like gap in treatment. Just to give you a sense, toggling back one. So this is like roughly the height of where these heaters are, and then below is kind of like a bevy of measurements. So there's seedling census, tree census, root measurements are taken, like soil measurements, soil respiration, soil methane, like many other samples. Okay, so one of the first things that is kind of like enormous finding from this experiment, and that has been, and the other thing I should mention about trace, sorry, is that it's the first ecosystem level warming experiment in the tropics. So we have kind of like analog results from warming experiments in other forests and in other tropical non-forest systems. But this is kind of like the first big time we've done this test in a tropical forest. It has been like, I feel like I can like brag about this because I have nothing to do with it. The amazing senior scientists who put this together and have been doing this for years. This has been like a labor of love. It has been like this amazingly successful project. And I feel so glad that I'm able to bring my undergrads to be a part of it. Okay, so one of the first big findings is that the soil respiration rates immediately took a huge jump in the warm plots. So broadly, warm plots are respiring much more CO2 than ambient plots. That ranges across time and across space. But even the most limited amount of extra CO2 emission is 42% higher than the control plot. And the largest amount is 200% higher. So like 3 times as large. So as you can see here, this is soil CO2 flux on the Y. This is the first year of the experiment. Blue is control. Orange is a warming plot. And you can see that broadly, like, we're talking about a really big change, right? Like from steady and low to super flashy, super respiry. And this has like potential long-term consequences, of course. So another really interesting result is these data are from like high frequency time series data from within the plots, but we were also curious about how space influenced this. So we did a survey chamber approach in which we walked around in the portable survey chamber and looked at CO2 emissions from over 30 spots across the entire, kind of the entire trace landscape plot level. This is by meters, so it's about 50 meters by 60 meters. And what you're looking at here is kind of a Bayesian spatial estimate of hot spots and cool spots of soil CO2 emissions. So warmer colors are kind of like higher emission areas, cooler colors are lower emission areas. And you can see there's a lot of variability across space. So one of the big takeaways here is just that it probably really matters. Like warming qua warming is not the only predictor, right? Like warming is probably going to interact with spatial heterogeneity in non-straightforward ways. Like you can even see that like this plot is, you know, shifting based on warmth versus control very differently than this plot. And we were able to kind of like quantify some of that across space so that we can kind of like feed this into models in such a way that, you know, we're never going to be able to have like 50 replicate warm plots. It's just way too expensive and way too difficult. So we're kind of interested in whether or not we can use these Bayesian spatial approaches to try to get a little bit better at saying, these are our manipulated control plots, and we have some of these relationship dynamics. This is what we know from a single time point across space. What do we think might be happening if you warmed up the entire landscape? And this is ongoing work too. So this is only the CO2 results, but we also spent last summer trying to track this down for the CH4 results. Those are kind of still in progress. I have a student who's probably going to be working on that this summer, but it's been like really fun to kind of like use some of these more cutting edge Bayesian spatial tools and these, you know, relatively newly accessible, easy ways to do a lot of spatial measurements quickly in the field because of new instrumentation, and kind of try to combine them to see if we can get better at predicting stuff across space. Okay, so that's kind of some of the results directly from the warming experiment. And I'll mostly shout out our 3 principals, Tana Wood at the US Forest Service, Sasha Reed at the USGS, and Molly Cavallari at Michigan Tech. They are like the gurus of this project and really amazing scientists and ecologists. But the follow-up questions that my students want to know about was kind of about this interaction between 2 different types of disturbances, acute disturbances and these chronic disturbances. So the chronic disturbance in this case is warming, but 2 of my students who wanted to pursue honors theses decided they wanted to track down what might be happening when that warming interacted with a more acute disturbance. So one of them, Nibia Becerra Santillan right here, wanted to know kind of the opposite of the drought question, which was when there's big dumps of rainfall, which we also are expecting to become more frequent in this forest, what might that do to methane and CO2 emissions across space? And how is that going to interact with a warming climate? This is a video of Chundam, who's just kind of like taking some samples. And just to give you a sense of the forest, because you guys haven't actually seen a photo post Hurricane Maria yet. We'll talk about Hurricane Maria in a second, but broadly this forest has gotten, between the data you just saw on the last slide and this data, much more shorter statured, much more light availability. It's super palmy, lots of pioneer species, or sorry, early successional species have come into the system. There's a lot of tree mortality after Hurricane Maria. So this is definitely like an ecosystem in recovery as well. Okay, so Nubia wanted to know, this is an ecosystem recovering from a hurricane, potentially being warmed, and also probably getting big dumps of rainfall. So she decided she wanted to run a rainfall manipulation experiment in which she simulated large dumps of rain. She took what we estimated over the last 30 years to be about a 90th percentile rainfall dump, and she simulated it a bunch of times and measured soil CO2 and methane for 5 time points in the immediate wake of simulating a rainfall event by like literally pouring water on the ground. So yeah, she found a bunch of really interesting things. So the first thing that she found, and I'll walk you through these results in a second, is that the methane, and I'm only going to show you methane results here, is that methane flux rates from soil varied not only based on the rainfall event, which we were expecting, because we have a pretty good understanding of how rainfall events influence soil methane flux, but also very strongly on underlying ecosystem heterogeneity. And one connection to broader global change impacts that Nubia wanted to make is she picked her locations where she did her rainfall manipulation based on locations that had been more and less impacted by Hurricane Maria. So what you're looking at here is control group. So she picked 2 locations. P1 is an area that was not very impacted by Hurricane Maria, relatively speaking. It didn't have a lot of tree mortality in that immediate area. It was relatively closed canopy by the time she did this experiment, and the hydrology hadn't shifted that much since pre-Maria. And then P5 was in the part of trace that was kind of like the most altered by Hurricane Maria. There's been a big change in hydrology there. We have a big nonlinearity in our soil trace gas fluxes in our time series, and plants are growing back really differently. So the control plots, she did not dump any rainfall. So you can see here, both of these locations had a net uptake of methane at her rainfall event, which didn't occur because this was the control, steady, 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 steady. Great. Meanwhile, her experimental group, she took a pre-fake rainfall soil flux measurement here. And then in the immediate wake of adding rain, so increasing soil moisture, driving down soil oxygen, there was a sharp decline in how much methane was taken up by soils, totally expected. And then that kind of steadied and recovered by 24 hours after the rainfall event. Cool, pretty straightforward story. However, the differences in heterogeneity ended up skewing kind of how the recovery looked over time. And it's a little bit easier to see in this graph. So what you're looking at here is a time series normalized across all 4 of the locations, 2 controls, 2 experiments. And I want to point out measurement time too. So this is the first CH4 flux measurement in the immediate wake of this big dump of rain. And you can see, and on the y-axis isn't the total amount of flux, it's the difference. So how extreme the reaction was in that spot to the exact same rainfall event. And you can see that P5 didn't have as strong of a reaction as P1, even though P5 is the spot that the hurricane really busted up. So one of our big takeaways here is this really impacted location actually didn't react as strongly to this rainfall event as a relatively stabilized post-hurricane location, sorry. So it's not totally a given how 2 subsequent acute disturbances might interact with each other, even in one location. And we didn't even do this in the warming plots. So that's actually something we're following up with this summer. It's really tricky to do these sorts of manipulations inside the warm plots and the controls because space is really limited. And it's not really an experiment where you can kind of like mess with things very easily without throwing off a bunch of other data. So we're kind of like thinking about a way to track this down this summer. So yeah, one of our big findings is that disturbance-driven heterogeneity might kind of like send ecosystems on alternate pathways in terms of how they react to future disturbance events. Okay, so to talk about Miriam Tleet's project, Miriam was really interested in Hurricane Maria more directly. And before I talk about kind of the ecology of her project, I don't really like to talk about Hurricane Maria without acknowledging that this is a real environmental disaster that affected real people. I think sometimes as scientists and ecologists, we can just sort of like, at least I know I can sometimes just have my head in the data and be thinking like, here's what I measured, carbon, phosphorus, nitrogen, whatever. But I'd like to acknowledge that Hurricane Maria led to the death of probably about 3,000 additional people in Puerto Rico. I wasn't on island during Hurricane Maria, and I don't know anyone personally who died, but I know people who lost family members, people who lost their homes, and the kind of like legacy of trauma from Hurricane Maria is very present still in Puerto Rico today. And I think something I try to bear in mind when doing this kind of work is whether or not I'm kind of honoring that legacy appropriately by kind of just like doing ecology. And I think that's been something that has been really difficult for folks who work in any really impacted ecosystem to think about how they're using their skills and their time ethically and morally. And I still don't necessarily think that I've landed on the right answer. Like there were moments after Hurricane Maria when I was like, I should have been a doctor, you know, or a nurse or like a public health professional or not a carbon scientist. But I think where I've landed now is that like sort of this is the skill set I have, and this is the way in which I engage with kind of our broader world. So bringing that to a system where I can do as much good as possible is kind of like my personal current moral imperative. But I'm also really open to talking to any of you about this too, because I'm sure you've kind of like reached some of these moral quandaries in your own work too. Okay, so with that, even though I just said, hey, it's really important to remember all the people that were affected by Hurricane Maria, I am going to talk a little bit about the ecology of Hurricane Maria. Okay, so this is a photo from one of our field sites before Maria. This is a day or two after. It's like pretty crazy to think about how drastically this ecosystem was impacted. And I'll highlight a few specific ways that we think about like the big biogeochemical impacts. So wind shear is what kind of like ends up ripping off a lot of this foliage and dumping it on the ground. So sometimes we think of this as basically like a large fertilization event. Because these aren't senesced leaves being dumped on the ground. These are like NMP rich, like very alive leaves. So there's a big debris input. There's likely after most of these hurricanes, a big nutrient pulse. There's a stream in this photo out through these streams. From previous hurricanes, we know especially NO3 minus and potassium are likely dumped out of these streams. There's also relatively strong data showing that carbon, phosphorus, and nitrogen stocks shift in the kind of like 1 to 2, but also 5 and 10 years after these big hurricane events, also probably down through the soil profile. Okay, so Miriam right here, Miriam Tleet, who's doing her honors thesis now, wanted to know how the changes to plant communities because of these disturbance events like Hurricane Maria would feed back to carbon and nitrogen cycling. So what she did was she took 3 exemplar tree species, 2 are early successional, Cecropia species and a Pistoia species, and 1 is a late successional species, a Guarea species. And she picked 5 of these trees, and then in the immediate 1 meter period around their stem, she measured a bunch of carbon and nitrogen variables. So she measured green leaves, senesced leaves, live roots, dead roots, soil microbial biomass, bulk soil, and greenhouse gas emissions. So basically your question is, we know that there's a zone of influence around different tree species that differs biogeochemically based on species. And we know that something like a hurricane is shifting what species are where. So her question was, hey, is a changing hurricane regime going to shift carbon and nitrogen in this forest? And this work is still ongoing, but broadly, we think that, yeah. So she found some pretty distinct differences in nitrogen cycle feedbacks based on whether or not you were close to the successional long-term species, the Guarea species. And this is the species, so relatively high in percent nitrogen in leaves. She also found that the soils right around these trees had a different nitrogen pattern than soils around these early successional trees. And we also know that this species is much more likely to be negatively impacted by hurricanes. And in fact, our seedling census data shows that Guarea are becoming less and less frequent over time in the system in the wake of Hurricane Maria. So I mentioned here that she found some differences in extractable carbon and nitrogen. So you can see that Guarea, these late successional species, have less extractable C and less extractable N in the immediate soil around them. And right now, what we're kind of like working up to is thinking about, you know, kind of like how we can best quantitatively model what that might mean going forward for carbon and nitrogen availability. Okay, so one of our other big takeaways, this second case study has been a little bit of like a dump of like, here's a bunch of small projects that we began over this last year. But we're really enthusiastic about kind of like bringing all these small ways of thinking about disturbance together to think about some big picture questions, which are just, hey, like, how should we be using our toolkit to acknowledge the fact that a lot of science is done looking at only one disturbance at a time, maybe two, but we also know that these ecosystems are going to be subject to shifts in many different types of disturbances, right? Like rainfall regime warming, both of those probably chronic, maybe also acute, like heat waves, acute drought. We also know that there's going to be things like fire moving through, not this system, but other systems, hurricanes, disease, et cetera. And thinking about these interacting drivers, especially when it comes to how spatial variability is going to kind of mediate the response to these interacting drivers, is probably a huge imperative and really interesting. So yeah, broadly, I would say the direction that we're going in now, and another place I'd love to collaborate if anyone wants to, is thinking about how we can best kind of measure and predict and model shifts in the carbon cycle and the nitrogen and phosphorus cycles based on the fact that biogeochemistry is being changed along many axes by many different drivers with many different timescales and lots of spatial heterogeneity. So maybe that's like too big of a dump, but like to me, that's a lot of the most interesting questions that I'm really excited about right now are kind of like in the gray area of like, oh my God, a lot of stuff is going on. How can we possibly tease it all out? I don't know, but I think it's really important that we probably do. Okay, I'll just for 2 minutes talk about one interesting thing about being at a primarily undergraduate institution, and then I'll open it up for questions. Okay, so the last question that I alluded to at the beginning is that a lot of my students, and a joy of being able to be at an institution that kind of caters to these early career scholars, is that we get to kind of like help facilitate their enthusiasm and their early career interests. And a lot of my students are really interested in positive climate solutions and how we can use ecology to advance that. So a lot of them, as you might imagine, are super interested in agriculture and land use. So in my PhD, I did a lot of like kind of like big data work, looking at the Amazon, did some remote sensing stuff, like lots of kind of like, you're the god of the Amazon, what would you do? Very hypothetical, like theoretical ecology. Lots of maps like this. But how I've taken that work at Macalester is kind of like giving students the license to run with these really small super applied questions. So these are two of my students who have now gone on to graduate. They're both environmental consultants, which is like a fit with what you're about to see. Shannon Hahn and Evelyn Harrigy. And they wanted to do a project with Macalester Sustainability and the landscaping office within Macalester facilities, because they basically both hated lawn. And they were like, we think Macalester should not have so much grass. It should have way more native plants or fruit-bearing trees. So they ended up taking on this very like activist science project where they wanted to know, like, what are some ways that we can convince Macalester basically to remove some lawn and put in more ecosystem service friendly landscaping cover. So they ended up doing like a really cool kind of like qualitative project with the head of landscaping where they walked around, they kind of identified these win-wins, and then they came up with kind of like a GIS solution where they were like, this is where we think lawn patches should go. Like, we're not taking away where kids play ultimate frisbee, but like that weird patch of lawn between this sidewalk and the library, like it doesn't have to be turf grass, it could be something else. And they got super excited about this. So they ended up using iTree to kind of model, if you took their kind of like red patches, which is where they wanted Macalester to change their landscaping structure. If you took all those changes and ran them through an ecosystem services model, they basically quantified all the benefits of doing this and wrote up a report. And ultimately, the school did plant an additional 21 trees and convert like 5,000 square feet of lawn to native plants. And this work is ongoing. Like, these two students have graduated, but there's like an ongoing plan to add 4 new rain gardens. It's like become this huge behemoth. And it all started out of these students being like, we don't want to be professional scientists. We want to do like activist science. And now they're environmental consultants. And we've also done some kind of similar work partnering with local sort of like super mission driven organic farms. And it's actually been like really fun doing this kind of science with like a totally different paradigm where it's not really hypothesis driven. It's sort of like action oriented. And it's a really good fit for Gen Z and how they want to approach their careers. And it's also like really fun. Like they push me to think about doing science a different way. And like they still do rigorous like quantitative outcomes that are legit and not like inappropriate or anything like that. But it's just like a really cool way to think about using our toolkit for folks who want to go in different directions with their career. So yeah, my big takeaway there is just that like we could probably use the science to do tons of win-win interventions, even if like me, you're not really on the very applied science side of things. Okay, with that, I would love to just say gracias a todos for coming to this talk. I'm also going to shout out a bunch of our collaborators. I would not be where I am today without the many lab groups who mentored me, specifically the Silver, Hobby, Foley, Brat, Pesco, Plasky, Powers, and Venturia lab groups. That's a mix of groups at Berkeley, Minnesota, and at McAllister. We have a ton of collaborating institutions. I've had so many early career scholars help me with this work and do this work themselves. And I'll give two more broad shout outs. One is that this work is currently being supported by an institution that's located on the ancestral homeland of the Dakota people, and that this work would not be possible without kind of the unheralded work of administrators, custodial staff, and support staff who underlie all the stuff, keep the lights on, keep me from locking myself out of my office, et cetera. With that, I think we have about 4 more minutes. I'd love to take questions. Thank you for having me. Yeah.
So that drought study,
Yeah.
is that something that you planned, or how did, I mean, it sounds. like
No.
Whenever you do a drought study, it never works.
No, totally.
So you just had that, you happen to have those monitoring instruments. in
Yeah,
place, and then
exactly.
and just took advantage of that.
Pretty much, yeah. So this was my postdoc, and it was like one of those things where, you know, in science, there's sometimes these moments that like change your career forever. So I went to my postdoc lab, and I was planning to do like hardcore geochemistry, which hadn't been my strength, and I had done a lot of data science in grad school. So I was like, okay, time to do like my lab work or whatever. And so we had this like whole thing with like microbial ecology, and it was supposed to be pretty much a like topography, hydrology, geochemical mechanisms project. And then this drought happened, and it was like, your postdoc's out the window. Your new postdoc is drought, which was like kind of a fit for me because I had come into the group as like the global change driver person who had like more of a data science background. So I think they were sort of like, oh, good, Christine wasn't that good in the lab anyway. Like, let's get her to do all the stuff she's good at. But yeah, pretty much it was one of these like opportunistic, like that project can't even occur anymore because the hydrology we were expecting isn't there. And so now I guess we're measuring this other form of hydrology, which is actually an enormous global change driver. Yeah.
And then one other question on that, when you measured phosphorus.
Yeah.
And you said that it was available, what kind of measurement did you do for available phosphorus, and what was the depth of the soil?
Yeah, so the depth I was showing here.
It seemed kind of, I mean, based on what I know about phosphorus.
Yeah, tell me more.
Pretty high levels of phosphorus.
Yeah, so this data is, sorry.
Particularly for a forest. I don't.
Yeah, totally. I mean, especially for a tropical forest. Yeah, I mean, we usually think of this system as phosphorus limited, but I think like this particular forest, there have been a lot of studies coming out in the last like 10 or 15 years that are sort of like challenging that rule of thumb that P limitation is like the only thing you have to think about in a tropical forest, that probably there's more dynamic co-limitation with nitrogen than we previously thought. That's probably true here. So yeah, these data are only 0 to 15 centimeters. We did take data down to a meter in 15 centimeter increments, so I guess down to 90 centimeters. And then we used like pretty standard, just like extraction. So
Standard, like.
yeah, we used the, oh God.
Fluoride,
Yes.
right?
Yes.
Eggs.
Yes, we used a Bray extraction. Yeah.
Those are very, that's micrograms per gram, that's very high. Those are really high.
Do you think they're that high?
Yeah, yeah, those are really, really high, aren't they? Yeah.
I mean, they do follow the pattern that you would expect with, the raise and slope and the valley.
Right.
Increasing, with the deposition, geology and stuff, but,
Yeah, well, just in case I'm like out over my skis, let me once we're logged off, let me pull up my old lab notebook and just make sure that I'm not, this was 2016, so it might be a little fuzzy. Yeah.
Yeah, the other thing is the effect of drought too is pretty dramatic.
Yeah, definitely. So slides I'm not showing here just for the sake of time is, well, a lot of what we think, I guess this is soil water climate, so I probably should have shown the slide, but basically we mostly think that this effect is being driven by changes to iron redox conditions. So basically oxygenating the soil, shifts the relative ratio of iron 2 and iron 3. And then we think that a lot of this phosphorus ended up bound onto newly oxygenated or oxygenized iron 3. Yeah.
Kristen, you also
Yeah.
have one of the graph about the organic.
Yeah.
Next to it. So that graph, the y-axis has the unit of concentration, but if you convert that to the abundance, gram per square meter.
Oh, yeah.
How does that change?
Oh, interesting question that I don't know the answer to, but I'm happy to share the data. It's available online.
I tend to think that the difference will become even larger.
Oh,
Because
yeah.
You said you have some accumulation of the organic matter during the drought
Oh, because you're saying per area or per?
per area.
Oh, sure. Yeah, although I guess I don't think we had major differences in bulk density across space, so maybe not, but maybe. Yeah, we should run that down. That'd be interesting to look at.
Other question about your, I was surprised by how much difference you saw in CO2 emission by warming up.
Oh my God, yeah, it's wild, right?
Wow.
Yeah.
And,
Especially this is nuts.
Yeah.
Anyway, go ahead.
because I hear, that, when people use Q10 numbers.
Yeah.
Then they usually assume true.
Right.
And then they even say that, Q10 will be higher in the holding environment. And
Yeah.
Become negligible. But
Well, actually, we have a result about that too. So this paper's in revision, and a big part of the revision has been figuring out how to communicate about Q10. So basically, for those of you not familiar, Q10 is if you warm 10 degrees, basically how much is whatever process you're looking at influenced by that. So the warm plots, the Q10 within the warm plots is smaller than the Q10 outside of the warm plots. So there's probably some attenuation of this effect. So in a heat jag in the warm plots, there's not as much of a bump in CO2 emissions, but the overall baseline is so much higher that even though these soils aren't then responding to further warming, they're still just dumping so much CO2, it doesn't even matter. So the cool spots are flashier and kind of more sensitive to immediate warming events, but the overall microbial metabolism is shifted so much, maybe we think, that it's just throwing everything off. The other thing we've been trying to run down is how much of this is based on changes in root respiration. And that's kind of unclear too. So in the warm plots, there's a higher density of roots, but per root unit respiration went down. But probably overall, there's more root respiration in the warm plots because there's just more roots there.
Is that in those figures? Does that include root respiration?
Yeah, this is everything. And I think what we've been struggling to figure out is how to back those numbers out.
Yeah.
Which is just tricky to do. Yeah, for sure. But yeah, this experiment's wild, really, really wild. The PhD student I mentioned before, Rachel Cruz Perez, she wants to install soil warming rods down a meter. Right now, the warming, it does still extend down to a meter, but it's attenuating across space or across depth. So she's wanting to pump up these lower values of warmth or these lower depths, how warm they're getting. And I'm honestly anxious about what she's going to find. I'm worried that we're just going to dump all this carbon out. These soils are super deep. They're super carbon rich. Another interesting thing that's come up in review that I'm not totally sure what to make of this is like these warm plots have so much CO2 loss, but they don't, have been unable to measure any reduction in CO2 stocks. Which I guess shouldn't be like that surprising if there's so much carbon, or sorry, not CO2 stocks, carbon stocks. These are really carbon rich soils, so like, and we're not that good at measuring them that precisely. So maybe it's like expected that we shouldn't be able to measure anything, but like, we're dumping out CO2 and we're not seeing any measurable reduction that's statistically noticeable. I don't know. So that like one big question is basically, where is this carbon coming from? And like, is there some threshold after which we're going to start like drawing down the amount of carbon, or maybe this is all like a litter effect and we're actually just drawing down the above ground biomass? I don't know. Maybe other people in this room have a thought about that.
I'm not surprised that it's tricky.
Yeah,
I mean, even in a system that's really homogeneous, like something that we mix
Right.
up with tillage, you still need to make a lot of measurements for a long time. before
Right.
you get anything that's kind of out of the-
yeah, totally. Yeah.
Does the experiment involve like that primary part?
Yes, very roughly. So they do like old school DBH tree census once a year. Yeah, and a seedling census too. And I think they've had one LiDAR Passover, but pretty recently, and I don't think any of the results are out yet. All right, anything else? Thank you again for having me. Anyone who wants to chat about working at a liberal arts